Okay, so picking up where we left off here, guys, with receptor-mediated endocytosis, okay? Remember that endocytosis and exocytosis are separate from what we call the big three, okay? The big three were osmosis, diffusion, and active transport. Those are the three primary methods by which cells transport material. Okay? Endocytosis and exocytosis are only about getting very large things that can't go through the membrane, that can't pass through it, into the cell. All right? That's why they all have to be enveloped, okay? Or conversely, if it's exocytosis, sent in an envelope and then expelled from the cell. All right? So this is just dealing with really big stuff. Right? It's not typical cellular transport. This is the acquisition of raw materials in a big way. All right? So with receptor-mediated endocytosis, okay, we got those receptors, okay, and you can see there's kind of these little red forks, okay, on this diagram here, and they're absorbing, or they receive the signal that, you know, this hormone or protein or whatever it is they were waiting for is here, okay, and so the cell will invaginate its membrane, okay, and kind of fold it in, right, so that it does this, right, and then it will essentially kind of pinch off that vesicle, right, and so you'll have this bubble that then travels into the cell that contains that particular protein or enzyme or hormone or whatever it was that it absorbed, okay, and it'll go directly to where it needs to go. Everybody with me there? Yeah? Okay. Now that's actually kind of a very quick way to get things where they're needed and get large amounts of them into the cell fast, right? Because obviously something very big is not going to pass through the membrane of the cell. That's the whole purpose of the cell's membrane, is to keep very large things from getting inside. All right, so like we said here, the substances that bind to those receptors are called ligands, but you're not really going to need to know that. Okay, they're usually proteins, okay, like hormones. So receptor-mediated endocytosis is usually transporting large proteins, okay, and other large molecules that the cell needs. Okay, phagocytosis is usually the ingestion of food. Okay, pinocytosis is always the ingestion of fluid, water. Okay. All right. So what I want you guys to do here now is answer those 4 questions, okay? And then we'll go over them, and then we're going to talk about the effect of cell size on cell transport in preparation for what we're going to be doing, okay, next week. Okay, so for question number 1, okay, we've got color blindness being caused by a person's cells being unable to produce certain proteins. So where does the problem lie? Where is there a malfunction? In the DNA. Okay? A lot of people at this point will say it's in the ribosomes. But if the problem was with the ribosomes, the cell wouldn't produce any proteins. All right? The problem here is that part of the person's DNA is coding for the wrong thing. Okay, that's what a mutation is. All right, it's not something that makes you be able to have little blades that come out of the back of your hand, okay, or turn green or whatever else the X-Men can do. Okay, it is simply a change to your DNA that changes the way your body builds things. If you are diabetic, part of your DNA that codes for insulin doesn't code for insulin. It codes for a protein that's very close to insulin, but it isn't quite right. Your body produces it, but it doesn't work. All right, and so the cells in your pancreas think they're doing what they're supposed to do, but they don't produce insulin. The rest of the cells in your body can't absorb sugars properly, and as a result, you have to take artificial injections of insulin. Okay, and it's the same for color blindness. If you are colorblind, part of your DNA that codes for a protein that works in the receptors of your eyes that code for color, called cones, okay, don't work right. Right, those proteins are simply not produced. They don't, or they are produced, but they're not the correct shape, and as a result, they don't do the job they're supposed to do, and you can't see red or green or blue or whatever. Okay, most common form of color blindness is red-green color blindness. Most of the time, you would go through life not knowing you were red-green colorblind until you took your driver's test. Right, and then when they give you they make you look at these things that are like a collection of dots, and some of the dots inside are different colors, and those different colors make numbers and letters and stuff, and so you look at these things and you got to tell them what letter you see. Well, if you're red-green colorblind, you're going to look at those dots and go, I don't see anything, they're just a bunch of dots. Right? And then they'll know that you're red-green colorblind, which incidentally is why stoplights are arranged the way they are. You got red on the top, and green on the bottom. Or, if they're arranged horizontally, red's always on the left, green's always on the right. You probably didn't notice that, but that's why. Okay, they're always arranged that way. You never see a stoplight upside down, right? Red's always on the top, and that's just in case people are red-green colorblind. Okay, Mr. Van Erve in our science department here teaches the other AP class. He is completely colorblind. You can tell if he gets out of the house in the morning before his wife sees him. Okay, because he'll just be wearing, like, he'll wear two socks that are different colors. Okay, and he'll think they're the same. All right, we had one day when we were back in the old building, he was wearing like a purple tie with a green shirt. I mean, and it just, he's, you know, he walked in, we're like, Jerry, what's going on? He's like, what do you mean? What? He's like, what's wrong with your tie, man? Like, what are you wearing that tie with that shirt for? What? It looks fine. Jerry. You could tell he got out of the house before his wife had seen him. And I mean, he thought it looked fine because he can't see. He couldn't tell. He thought that the green tie he was wearing was a different color. Okay, I don't know what color he thought was going to go with the green shirt, but okay, like it didn't go. But he doesn't know that. Okay, he can't see the differences in the colors, and sometimes it's actually quite comical. And of course, we tease him incessantly about it. Okay, but it does happen. All right, and a lot of people don't even realize that they're colorblind until later on in their life. It's just not something they notice. All right. Everyone follow me there? Okay, so that's a genetic. There's lots of genetic disorders that work like that. Okay. All right. Number 2, why is it important for organelles to have their own membrane? So why is there a membrane around the lysosome, around the mitochondria? Why is that good? Andreas? Right. Okay. It gives them their own specific environments, okay, outside or inside of the cell where they can operate most efficiently. Number 3, what might happen if the lysosome's membrane ruptured? Well, we kind of talked about that in class. We said the digestive enzymes would escape into the cell, and then they would, of course, digest all the cellular parts and probably kill the cell. All right. What problems might a cell have if the ER was removed? Not that you could do this, okay, but what would happen? Okay, and they might not be able to do that at all, right? Because the smooth ER is responsible not just for transporting the lipids, but also for producing them. And essentially then, without the lipids, all the membranes in the cell would eventually break down, right? The cell might also have toxins build up inside because one of the other jobs of the ER is to detoxify poisons. To produce hormones, okay, all those kind of things. It does lots of stuff. Wouldn't be able to break down complex carbohydrates. There would be huge problems if your cell didn't have any ER. It would quite simply die, and it would simply die in short order, right? Because that's one of the most essential organelles there is. All right, questions on any of those? Okay, now quick thing here about cell size and cell transport. Just going to quickly touch on that, and then we're going to talk about the cell anatomy lab here. Okay. The bigger a cell gets, okay, so here's a big cell, here's a small cell, okay? How do cells transport stuff inside? What process do they use to transport things from place to place inside the cell? Peter? Diffusion, okay? Diffusion is the only way that things get transported within the cytoplasm. Okay, simply by diffusion. So where it's concentrated, it moves away from where it's concentrated to where it's less concentrated by diffusion. So if I have a cell that's this big, okay, nucleus is in the middle, okay, and we got, you know, this organelle here and another organelle there, okay, food particles, let's say this organelle is a lysosome. Food particles can only enter through the membrane. So they have to travel from here to here, or from, let's say, here to there. In this cell, here's the nucleus, same size. Here's that lysosome. Here's that lysosome. Food particles can be absorbed here. Which one of these cells is going to be more efficient? The little one. Why? Exactly. It takes way less time for nutrients to get to the organelles in the smaller cell. Because these cells have to transport things by diffusion. They don't have any choice. That's how they do it. It's going to take a lot longer for that material to diffuse to here. And maybe it doesn't diffuse to here. Remember, diffusion goes, it's really concentrated here, so it kind of spreads out. Okay? Maybe it doesn't even get to there before it's so dilute that it's no longer useful. Whereas if that process is happening in this cell, okay, here's where it came in. Well, it's still pretty concentrated by the time it gets to that organelle. You guys follow my reasoning here? Okay, so it's a big deal. If you're a big cell, transport is going to become a problem for you within the cell because you only have one choice for how to do it, and that is diffusion. All right? And the same principle is going to apply to getting rid of waste products. It's going to take a long time for waste products or toxic materials to diffuse to where they need to go, like the smooth ER, or maybe to the membrane to be gotten rid of, right? Those kind of things. Well, the longer a toxin is in the cell, the worse that is for you. Agreed? Okay, and your waste products, whatever, will maybe start to build up in the cell, and that's not good. That would be kind of like if you or I was trapped in a sealed room with no air supply. Eventually, our waste products, that is carbon dioxide, would become toxic. Okay? And that's the same thing that can happen to a cell when it begins to get very large. Okay? Cell transport becomes a problem, which is usually why a cell divides before it gets this big. Okay? They kind of have a sense of, size, and they'll divide before they become too large, right? But it's no different than trying to transport materials from one place to another in a big city. All right? I mean, if you look at Calgary, right? If you want to transport something within Calgary, you really have two choices: by train or by truck. All right? Now, if you're doing that by train, how many rail lines are there through Calgary? I have no idea. There's maybe 3, okay? Maybe 3 different rail lines or something like that. And there's only Deerfoot Trail, Stony Trail, McLeod Trail, okay? And across you got Stony and, let's say, Glenmore and 16th and Country Hills. Okay, those are really your only choices, right? In the same way that diffusion is the only choice for a cell. Well, it's going to take a long time if those are my only choices to get places in a city that's this big. Doesn't take very long to transport stuff in Okotoks, provided you hit some lights if you're very lucky that day. Right? But even still, even if you hit every red light in town, which is every day, okay, you still would be able to get from one side of Okotoks to another a lot faster than getting from one side of Calgary to another. Agreed? Okay, so that's kind of what we're going to be looking at on Tuesday, okay, after the cell anatomy lab. Questions there?